Hello, everybody, and welcome to our gene technology chapter for honors biology. So let's begin with our lecture today. All right, here we go. Now, we just finished a unit on DNA, and you learned about DNA, how it functions, what it does, how important it is. Well, this chapter is going to take DNA and we're going to apply it to other things. You probably have heard about forensics, where they're using DNA to identify people. We're going to talk a little bit about cloning on how they produce an actual genetic duplicate. So, this chapter is going to involve the technology that utilizes DNA. All right. So let's first begin with some concepts. No two people are alike genetically except identical twins. Fraternal twins are not genetically identical. Identical twins have the same sequence of their DNA, so they are genetically identical. About less than 1%, about 0.10% of your DNA varies from you to your neighbor, the person next to you. People look at them, they're like, wait a minute, we don't look the same. We're very different than each other. I don't get it. It doesn't make sense to me. Well, you and your neighbor, you and the people around you are producing proteins. A lot of the proteins as we learned in DNA chapter, a lot of the proteins that you're producing are the same, which means even though you have some physical differences, that way you look, a lot of your enzymes, your proteins inside of you are the same. They function the same, which means you have the same coding to produce those actual proteins. Um, Basically, what we're doing when we're trying to identify DNA, and this picture over here shows you what's called a DNA fingerprint. And by just glancing at it, you look, this is like the DNA fingerprint from the crime scene, you could say you could find it. And then you look at the crime scene DNA and you start looking for matches. And look, suspect number 2 matches the DNA fingerprint. So you would assume that the crime scene DNA matches suspect 2. So you have some connections with your evidence. All right. What are they doing when they're comparing DNA of two people? Well, in order to make DNA fingerprints like this one, you're looking at that 0.1% that's a little different. So we're going to be looking at the slight differences in our DNA so that we can create fingerprints that are slightly different from each other and that we can evaluate. They don't just use this for crime scenes. They can use it to find parents, who's the father, who's the mother, for example, origins, human origins, you name it. Okay. Now, in our DNA, most of our DNA is what's called non-coding DNA, which means it does not produce proteins, non-protein producing DNA. Now, how much of that non-coding DNA? There's some slight differences in our non-coding DNA from person to person. Okay, what is different about our non-coding DNA from person to person is these things called length polymorphisms. All right, so what we can do is we can look at a few different people. Here's a picture showing it to you. Let's pretend this is person 1, this is person 2, this is person 3, and this is person 4. All right. What we are realizing is that each person has, and these pink areas are what's called VNTR regions. And I'll explain to you what those mean in a second. What you have in your DNA, in this non-coding DNA, you have repeating nucleotide sequences, such as, for example, CA, 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 and they repeat for a certain amount of time. They keep repeating CA, 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 CA. All right. Now, they sometimes occur in tandem. They occur in different, you have different amounts of them. For example, this is showing you 2 of them. This is showing you 3 different repeats. This is showing you 4 repeats. This is showing you 5 repeats. So what do we do with these things? Well, what they do is the area between your genes varies in length. So for example, you and your neighbor have 98% of the same DNA. Okay, 98% of our genetic makeup is, 98% of it is non-coding DNA. We have 0.1% of our DNA is different. So we have almost all the same genes. So how can I identify person A from person B? Okay. How can I identify the few people from each other? The way I would identify them using a DNA fingerprint is I would take their DNA, I would cut it up in pieces, and their pieces would be different lengths. Why are their pieces different lengths? Their DNA is different lengths because of these variable number tandem repeats. They have repeating sequences in their DNA, and some people's are longer and some people's are shorter. So when I cut your DNA into pieces, you create different pieces of different lengths. Now I'm going to go back to this. These lines here represent pieces of DNA of different lengths. And why are they different lengths? Because of these repeated subunits, these repeated units, these VNTRs, and each person has a different amount of VNTRs. Okay, so it's showing you might have 2 repeating units, you might have 3 repeating units, you might have 4 repeating units. So then we cut up these people's DNA, and let's say all these people's DNA gets cut here, and one person's gets cut there, one person's gets cut there, one person's gets cut there, one person gets cut there. So it shows you that because of these repeating units, the length of their DNA is what's different. And that's what a length polymorphism is, variations in the length of the DNA molecules. Okay, so our DNA is different length. If we take DNA and we cut it up into pieces, and these arrows are showing you where the DNA is cut, we will have pieces of different sizes. That's what makes you different than your neighbor. So I have to cut it up first, and then I can analyze the pieces of DNA. Okay, pieces of DNA after we cut it up are called RFLPs. These are fragments of DNA. They're cut by these things called restriction enzymes, and RFLP stands for restriction fragment length polymorphisms. What that means is you cut DNA into pieces into restriction fragments, and those restriction fragments are different sizes, different length polymorphisms, depending on how many of these repeating units they possess. Now, when you're trying to identify DNA, we're going to be real general in this first time we discuss it. And then during our lecture, we're going to break down some of these processes in more detail. The first thing you got to do is you got to first find the DNA at a crime scene. Usually, let's just say you're looking for DNA at a crime scene and you find it. Then that's this process of isolating it. Okay. A lot of times when you find DNA at a crime scene, you don't find that much. So you need to make more of it. And this process called the polymerase chain reaction amplifies or makes more DNA. Okay, so if you amplify DNA, you don't make it bigger, you make a lot of it. You make more of the same thing you have. Then you take DNA and you cut it into pieces. I just described to you, if we cut DNA into pieces, we have different size pieces. That's what makes us different than our neighbors. It's how long our DNA is, okay? So and what do we cut DNA with? These things called restriction enzymes. Then we're going to learn later in another lecture about how we sort the DNA. Well, to sort the DNA, we need to sort it by size. We use this thing called gel electrophoresis. It gives us a visual of what our pieces of DNA look like, and I already showed you one in the very beginning of the lecture. And then we compare the sizes of the pieces to a known sample, and then we can analyze that. Basically, we can go back to this example, and we know who is at a crime scene. So these are pieces of DNA that they found at the crime scene. This is the suspect's DNA pieces. This is suspect 2, suspect 3, and suspect 4. Since suspect 2's match, we're assuming suspect 2 was also at the crime scene. Now, the process of amplifying or making more DNA is called polymerase chain reaction. We learned in a previous chapter, DNA polymerase is used in DNA replication. So what is the polymerase chain reaction? It is a reaction that we figured out how to do in a laboratory of how to take DNA and make a lot more of it, replicate it over and over and over again. Why would we want to do this? So that we have more of it to work with, basically. A lot of times you don't find a whole lot at a crime scene. You might want to run many tests on it, so you need to make many, many copies of it. In order to do it, you need 4 things. You need DNA nucleotides. And by the way, the machine, it's not that big. It's a small little machine. It's like this big. And this little machine, this big, you can take this little machine and you can make a bunch of DNA in it. Okay. It's not even a crazy tool. It's not this huge lab apparatus or anything. It's a small little mixing device, and it mixes it and creates the right temperatures for it to happen at. So the polymerase chain reaction is a chain reaction. It happens, then it happens again, and then it happens again. So you start with, let's just say you start with 1 DNA molecule, you end with 2, then you do it again, 2 become 4, 4 become 8, 8 becomes 16, and it keeps doubling. They use the polymerase chain reaction. That's one of the tests they're doing to test for COVID. So if they find pieces of DNA, they swab your throat or swab your nasal cavity. They try to extract some DNA from the virus by doing that. And then they throw that, whatever they find, in the polymerase chain reaction, and they try to make a bunch more of it. And if they find it, then if they can make it, it means they found it, basically. So that's how they detect it. It's very sensitive because it doesn't take a whole lot of DNA. Start with a little bit of DNA, and you can make a whole bunch out of it. So it's a very sensitive test. To make DNA, what do you need? You need the template, the beginning, the DNA that you're going to actually try to amplify or copy. You need nucleotides, A's, C's, T's, and G's, so that you can build new DNA. You need the enzyme. Remember this bad boy, this enzyme? The enzyme that makes our DNA replication enzyme. The main one that adds nucleotides is DNA polymerase. And then you need what are called primers. which are, if you probably remember from DNA replication, they help begin the process of producing new DNA. So what happens? You start with your DNA. Okay, here's the first steps. You have your DNA. This is your template. And then this machine I was telling you about heats the DNA. Heating the DNA breaks the hydrogen bond. So now you have two completely separate strands of DNA, completely separated from each other. The heat separated them from each other. Okay. Step two, it cools. When you cool it, it allows those things that you added called primers. Primers begin the process of replication. So primers attach. Primers attach. Okay. And primers are basically single-stranded pieces of DNA or RNA. We learned in our last unit that RNA was used, but DNA is what these are going to probably be made out of. And what do? They attach to a beginning point and they begin or initiate replication. So a primer lands there, a primer lands there. Okay. Then DNA polymerase, that enzyme, remember that DNA polymerase enzyme comes in, and what does it do? It adds nucleotides from the 5 to the 3' direction, 5 to the 3' direction. When it is done, you have two new, so one strand of DNA became two strands of DNA. Then the polymerase chain reaction, it does it again. That's why it's called a chain reaction. It does it again. Then 2 become 4, and then 4 become 8. Then 8 becomes 16, and then it keeps doing it. I believe when they test for COVID, they swab your throat, and I believe they run it through a PCR test of over 30 cycles, which means they double it 30 times. So if you had very little virus left, and the virus has DNA in it, if they had very little viral DNA left, it could take, and let's just say you don't even feel symptoms, it could take a very small amount of viral DNA that doesn't even give you symptoms, and it can let you know that you have a positive test. You may not have symptoms, but you did have the virus inside of you if the DNA was present. And here are the PCR steps. These are the steps 1 through 4, and I explained them to you using that picture a second ago. So once again, you add your template, you add your enzyme, you add your nucleotides, you add your primers to begin the process, you heat it, and then the heating separates it, okay, separates the strands, and then you cool it, the primers attach when you cool it, and then DNA polymerase starts adding nucleotides in the 5 to 3' direction, and it does it again, and then it does it again, and then it does it again. All right, so restriction enzymes, I've briefly mentioned them. These are DNA scissors. They cut up DNA. They're DNA scissors. What did they do with DNA? Where did we find them? What do we know about? Well, we discovered them in bacteria. Scientists discovered them in bacteria. I just explained to you how we test for COVID, and COVID is a virus, and viruses are made out of DNA or RNA in a lot of cases. So let's just assume they're made out of DNA to make this simpler. So if a bacteria gets infected by a virus, it needs to defend itself, because what does a virus do? A virus basically invades the bacteria, just like you and me. It'll invade the cell. It takes over their DNA, it takes over their information, their coding, and changes it so that it benefits the virus. And in a lot of cases, it hurts the cell. So just like you and me, bacteria get infected by viruses. Actually, more bacteria are killed daily by viruses than viruses kill humans or anything else on the planet. So viruses attack bacteria. So what do bacteria do? They release these things called special enzymes called restriction enzymes. And what do restriction enzymes do? They cut up the DNA of the virus, getting rid of it. They kill it. They cut it up into pieces, and they make it not able to infect them anymore. Now, when you cut DNA, sometimes DNA gets cut in a staggered fashion. If you notice, this got cut in this fashion. It came down here, did like a stair step, and came over, and it formed this thing. See how there's these bases that are unpaired, and these bases down here they're called sticky ends. Sticky ends, which means this type of technology, when you're dealing with restriction enzymes, this type of technology can be used to cut DNA out of something and insert it into something else. So this type of DNA is very important in genetic engineering and things of that nature. Now, basically, these are pieces of DNA. So if we cut this DNA, we have two pieces of DNA. That means we have two RFLPs, RFLP1 and RFLP number 2. These are pieces of DNA after it's been cut by a restriction enzyme. Question here says, how many base pairs does each RFLP in the picture? So whenever we talk about how big a piece of DNA is, we're going to be talking about how many base pairs or BPs does it have. So these are not paired up. They're no longer paired up. So RFLP number 1, I would put in the answer here, RFLP number 1 has 2 base pairs. So your answer for his, 2 base pairs. RFLP number 2, 1, 2, 3 base pairs. These are not paired. These are not paired, so they do not count. This is the answers, 2 and 3. Now, how do you use restriction enzymes to diagnose disease? It's actually really simple. My answer to you is you should write somewhere on this sample in your notes. Write the 3 words. Step 1, cut DNA. Step 2, count the base pairs. Step 3 should be compare numbers. So you'll have numbers down here. So step 1 is cut. Step 2 is count base pairs. Step 3 is compare. 3 C's: cut, count, and compare. So what you do is you have a mutated gene and a healthy gene. So this is good DNA. This is bad DNA. Okay. Now we will give you information. Like, for example, I said there's a restriction enzyme called VAL. I named it after Valhalla, and it recognized the following DNA sequence. GGTAC is a sequence it recognizes and cuts between the T and the A. So this is how it cuts. GGTAC, it cuts straight down between the T and A. Okay, so here's the, so this is given information. You don't need to figure it out. I will tell you this information. Okay. Now, these diseases we are going to be diagnosing, just so you know, these are recessive diseases. So if someone is homozygous recessive, like 2 small a's, they are diseased. If someone is capital A, they are completely healthy. They are homozygous dominant. We would say they are totally healthy. And once again, and finally, a person who has a good and a bad gene is their good gene covers up their capital A covers up their lowercase a, which is diseased, and they are heterozygous, so they are healthy, but they are carriers, which means they don't have these diseases. An example of this would be like sickle cell anemia. They do not have the disease. They are healthy, but they carry the bad gene, which means they could give it to their baby. So your 3 choices are going to be a healthy person, a carrier who is healthy, or a diseased, completely diseased person. So I told you, step 1 is label the cuts. Your DNA should look like this. GGTAC, I cut it. GGTAC, I cut it. GGTAC, I labeled the cut. Over here, it's GGTAG. That does not match up. That is a C, that is a G. It's not a perfect match, so there's no cut over here. It needs to be a perfect match, okay? So we've just done step 1. I said step 1, label the cuts, okay? Cut the DNA and draw it in on your DNA, okay? Step 2, count how many base pairs there are in each piece. And by the way, these restriction enzymes aren't making a staggered cut like I showed you before. They're making a straight cut. So you count the base pairs. 1, 2, 3, 4, 5 here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 here. You got this one is 5, this one is 15 across, and this one is 6. All I'm doing is I'm counting the pairs. 1, 2, 3, 4, 5, 6, and so on. If you count across, you'll get 15. So we've done steps 1 and 2. Step 1, cut the DNA, label the cuts. Step 2, count the pieces, the base pairs and the pieces. And then step 3 is compare the numbers. If you look down here, if someone, fetus A, has pieces, RFLPs are the pieces, they have a piece that's 5, a piece that's 6, and a piece that's 15. Well, 5, 6, and 15 are all matching a healthy person. So fetus A, healthy. Homozygous dominant, healthy. Okay? Fetus B, this is if we're testing an unborn fetus. We want to see if the baby's going to have the disease. Fetus B has pieces 5 and 21. If you discovered that, you'd be like, wait a minute, this is our mutated gene. This is not good. This is our disease gene. So if a fetus has pieces 5 and 21, you would be telling them that they are going to be diseased, or the parents of that fetus that they will be diseased, because it matches 5, 21 match perfectly with mutated. If you notice, fetus C has pieces of all kinds. It has a 5, it has a 6, it has a 15, it has all the pieces of DNA, which means fetus C has a healthy gene and an unhealthy gene. So fetus C would be what we would call our carrier. And here are the answers for you there. Okay, so healthy, because 5, 6, and 15 match 5, 6, and 15 match healthy. 5 and 21 diseased, and 5, 6, 15, and 21, all the numbers means you have a diseased and a healthy gene. So this would be our carrier down here. In reality, you cannot just count how big a piece of DNA is. So in reality, we need to visualize our DNA. So I'm going to show you how we visualize the DNA. This picture, this is called, you're going to learn about in our next lecture, it's called a gel electrophoresis. So what does it do? It separates the DNA by sizes. So for example, person A had pieces 5, 6, and 15. Well, look, this is patient A. So this is kind of like a number line. This first row is our comparing row. It's like our baseline. So we know that each piece lands here, it's going to be 30. If a piece lands here, it's going to be 2. If a piece lands here, it's going to be 4. If it's 6. So notice how this one's between a 4 and a 6. This is 5 base pairs. This one is 6 base pairs. This one here is between 14 and 16. This one's 15. So this one's 5, 6, and 15. 5, 6, and 15. 5, 6, and 15. So you're using your number line here to compare how big each piece. These lines represent the pieces of DNA. Okay? Patient B has a 5 and a 21. There's their 5, and there's their 21 right there. Patient C has a 5, 6, 15, and a 21. There's their 5, there's their 6, there's their 15, and there's their 21. So this is how you would actually visually see the pieces of DNA and how big the pieces are. So the example I showed you, we cut, we count, we compare, but in reality, we're going to be comparing to something like this. All right, that should end our lecture number 1.